Hi everyone, this is Dr. Hall, and we're going to do an introduction to the histology of the reproductive system, or the things we can see through the microscope. So I want to start first with a review of the structure of an ovary. So the ovary has an outer region called a cortex. So this kind of outer region, much like we had a renal cortex. So this outer region of the ovary is called the cortex, and it is there that we're going to find the follicles. So the follicles are groups of cells that surround the developing oocyte or egg cell. And so there's this ovarian cycle. There's a sequence of events that occur as the oocyte goes through its maturation process along with the follicle, which also matures, eventually getting to ovulation. So to start, each month some immature follicles, which are these smaller follicles that we see over here, are going to be recruited to start maturing. So those follicles are going to enlarge, the oocyte is going to get a little bit bigger, and then there are going to be more and more cells surrounding the oocyte. So you can see now this one has even more cells, and then eventually we start to get some fluid building up in the center of the follicle that pushes the oocyte to one side. Ultimately, this will become quite large and actually bulge off of the surface of the ovary, at which point it's called a mature or a Graafian follicle. And that'll have the oocyte within it, located right here. So in the middle of the menstrual cycle, then there's going to be a positive feedback loop between estrogen and LH that's going to cause that mature or Graafian follicle to rupture and release the oocyte with its surrounding halo of cells here. Here's the oocyte out into the pelvis, where it hopefully will be slurped in by the fimbriae into the fallopian tubes, where the ciliated columnar epithelial cells will wave their cilia and waft the oocyte into the fallopian tubes and then down toward the uterus. Now, after ovulation occurs, the follicle's job is not done. So that empty follicle then is going to turn into the corpus luteum, which is Latin for yellow thing, because literally when they cut open ovaries, they were like, oh, what's this yellow thing? But it sounds much fancier in Latin, right? So corpus luteum. The reason why it's yellow is because those cells of the empty follicle start making the hormone progesterone, and that's what's responsible for the significant buildup that we see in the endometrial layer of the uterus, because we're hoping to get pregnant. So preparing that endometrial lining so that a fertilized egg can implant into it. If there is no pregnancy, then the corpus luteum will give up and scar down into a white thing, which in Latin is corpus albicans. Progesterone levels fall, and then the endometrium deteriorates, breaks down, and is shed as a menstrual period. So this is kind of the ovarian and menstrual cycle that occurs every month. In this diagram, they have chosen to kind of show these different stages around the ovary just so that they can show you everything all in one diagram. So I just want to point out that these follicles are not actually moving around the ovary. They're staying in the same place. They're just showing us several different follicles in different stages for the purposes of education. So let's take a look under the microscope. So at low power, we can see around the edge of the ovary, right? So in the cortex of the ovary, around the edge here, we see these little tiny circular things, right? So this is on very low power. This is probably on 4x or 40 total times magnification. And so if we come in a little bit higher power, we can see here is an immature follicle. Here is an even less mature follicle, right? These are even smaller. So these would be what we call primordial follicles. And this one up here has gotten to the stage where it is a primary follicle. So the oocyte starts to grow and mature. The cells surrounding the oocyte that are the follicle start to grow and mature. And then eventually we're going to have multiple layers of cells surrounding the oocyte in that follicle. So here is an example of a follicle that is kind of in between immature and mature. So this on the left, we have very low power, on the right, somewhat higher power. And so we can see this whole thing is the follicle, and all those little purple dots are individual cells that are surrounding the oocyte. And so our oocyte here is this guy right in the middle, or gal. And then we can also see in this particular example that the fluid is starting to accumulate in that antrum, or that space. We're starting to get a fluid-filled space, so this one is getting close to being fully mature. Here is another image where we can see here up above, this is very similar to the one we were just looking at, might even be the same one, I can't remember from when I took these, but that is getting close to being mature. And then we see this big one right here, and this is almost fully mature. So we're going to call this a mature or a Graafian follicle. We have our oocyte in the center surrounded by some follicular cells, but then most of the follicle is filled with fluid. Eventually the oocyte will come to be against one side, and the entire middle of the follicle will be filled with fluid. That is a mature or Graafian follicle. It will bulge off the surface of the ovary and then burst due to that positive feedback loop between estrogen and LH and release the oocyte with its surrounding cells. Here's just a couple of other examples. So again, we have a close to mature follicle here, the mature follicle here. You can see those oocytes in the middle. And here is a close-up, very high-power view of that oocyte or egg cell. And it looks really grainy, and that's because the chromatin is all unwound because it's busy working on meiosis. So it's a pretty busy cell in there. Okay, so that's what we're going to see when we look at slides of an ovary under the microscope. What about the testes, also called testicles? So these two words are used interchangeably. I apologize. Testes is easier to say. It's shorter. All right, so a quick review of the anatomy of the testicles. First of all, we have these little lobules, these little groupings of squiggly tubes. Those squiggly tubes are the seminiferous tubules. They look kind of like ramen, and that's where the sperm are going to be produced. We're going to take a closer look at those in a moment. This area here, which is this coiled tube that sits on top of and curls around behind the testicle, is the epididymis. And then we can also see in this diagram the pampiniform plexus of veins, as well as the testicular artery and the vas deferens and the testicular nerve. So lots of things we can see in that particular diagram. So if we were to pull out one of those seminiferous tubules and take a cross section of it, we would see some really interesting things. So first of all, around the edge of the seminiferous tubule, you're going to find the spermatogonia. Those are the precursor cells that will undergo mitosis, make exact copies of themselves, and then some of those extra spermatogonia are going to start the process of spermatogenesis. So that will be that process of meiosis, right, where we're going to end up taking one cell and create 4 cells out of it, each of which is haploid, or N, only has 23 chromosomes each. The other accomplishment of meiosis, besides having the chromosome number and giving you 4 gametes, is that it also allows, through that crossover step, for kind of the mixing up of the combinations of genes present on individual chromosomes, so that those 4 gametes are going to be all genetically different from each other in terms of which gene combinations they have. So the spermatogonium starts out around the periphery of the seminiferous tubule, and then as these spermatocytes, as we then call them, start to undergo meiosis, they migrate towards the center of the tubule. So as they are doing this process, they gradually move toward the center. There's a couple of other cells that are important. One is the Sertoli cells. So the Sertoli cells are these kind of long, skinny cells, and they span the whole distance that those spermatocytes are going to be traveling as they mature and undergo meiosis, and they support and nourish those spermatocytes on their journey. The other cell type you're going to see are the Leydig cells. Those are located outside of the seminiferous tubules, and those are the cells in the testicles that actually make testosterone. So that's pretty cool. So Sertoli cells support and nourish developing spermatocytes, and the Leydig cells produce testosterone. By the time these spermatocytes get to the center of the tubule, they are either spermatids or spermatocytes. So they are going to shed their extra cytoplasm and organelles, slim down, get that head of a sperm, the midpiece, and the tail, and then finally they will move into the epididymis to mature and await ejaculation. So when we look at the testes underneath a microscope, even on low power, we can see these are all these cut cross sections of these tubules. Right, just like the diagram that we looked at. This one we kind of caught on oblong a little bit. This one we caught a little bit oblong, right? But so there are all these tubules, so we can kind of come into slightly higher magnification and even higher, and now we can see all kinds of interesting things. So around the outside, these really dark cells here, these are our spermatogonia. These are the cells that are going to make exact copies of themselves via mitosis, and then those copies are going to turn into spermatocytes and undergo meiosis and keep moving toward the center of the tubule as they undergo that process. We're also going to see Sertoli cells. They're pretty difficult to see, but they're kind of these long, skinny cells that go the whole length from the outside of the tubule towards the middle there. They're supporting and nourishing the spermatocytes. And then, of course, we can see Leydig cells outside of the seminiferous tubules. So it's pretty neat to see all the different components of those seminiferous tubules in the testicle. And if you have any questions, please let me know.